Good evening, everyone. On the behalf of ShareGenes Group, I warmly welcome you all to the lecture number 4 of the ShareGenes Knowledge Lecture Series. Today's lecture is particularly special. For decades, scientists searched for nature for the perfect genome editing nucleases. Today, the artificial intelligence can design a one from the scratch. So this is the story of the Plant-OpenCRISPR1, the world's first AI-designed genome editing platform for the plants. It is a landmark innovation demonstrating now how AI is transforming the way we engineer the plant genomes for the sustainable agriculture and global food security. We are truly honored to have the scientists behind this pioneering innovation with us today. I hope this session inspires new ideas, meaningful discussions, and future innovations. So I now invite Dr. Anil Kumar, sir, scientist at ICR Central Rice Research Institute, Cuttack, to formally introduce our distinguished speaker, Dr. Kutubuddin Ali Molla, sir.
Hi, everyone. This is Anil Kumar. It's my pleasure to welcome you all to today's seminar. It's also my honor to introduce our distinguished speaker and my dear colleague, Dr. Kutubuddin Ali Molla. I believe many of you already know Dr. Kutub. He is a senior scientist at Central Rice Research Institute, Cuttack, and is internationally recognized for his pioneering contributions in genome editing. More specifically, he is a leading developer in innovative genome editing tools. He has developed a compact genome editing tool called TNPB, which is an alternative to the widely used Cas9 system. He has also developed Plant-OpenCRISPR1, an innovative platform that integrates the AI with genome editing. I'm sure he will be talking more about this in today's lecture. Beyond his outstanding scientific contributions, Dr. Kutub has been an inspiration to many of the young researchers. Alongside his high-impact research, he is deeply committed to mentoring and nurturing the next generation of scientists. His scientific journey includes a prestigious Fulbright Postdoctoral Fellowship at Pennsylvania State University in USA. He has also received numerous prestigious awards. A few of them includes INSA Young Scientist Award, INSA Associate Fellowship, Young Scientist Award from Indian Science Congress Association, and also he is a selected member of Indian National Young Academy of Sciences. There are many more awards in his cap. Today, we are privileged to have Dr. Kutub with us to share his insights on genome editing with more special focus on integrating AI with genome editing system. Please join me in extending warm welcome to Dr. Kutubuddin Ali Molla. Sir, please start your session.
Oh, thank you very much. Please let me know if I am audible and in right sense, or should I be louder, or it is okay? The voice is okay.
Yeah, it seems good.
Good. Okay, great. So thank you very much, Anil. Okay. So thank you very much, Anil, for introducing me to ShareGenes platform, and it is my privilege to present this work to all the participants registered today. And I think I should congratulate ShareGenes on, building this platform to share the cutting-edge science that is going on around genetics and plant breeding. So that's a great initiative, and I must appreciate the mind behind this ShareGene platform. And Anil, thank you very much for your kind introduction. And Anil is also a Fulbright, so we are kind of alumni of Fulbright. So he just completed his Fulbright from Minnesota and came back to our institute. So he's a great colleague of me. Okay, so is it okay? PPT is visible? Anil?
Oh, not yet, sir. Yeah, it's coming up.
Okay, great. Wonderful. If you see any kind of, misconnection in between, so you can just knock me. So because I cannot see you, I can only see my slide while presenting. Okay.
Yeah.
So if there is any problem in audio and video, you can just knock me. Okay. So yes, as Anil said, that I'd be talking about today mostly on this recent development from CRRI, Central Rice Research Institute. That is on Plant-OpenCRISPR1, and that has a huge potential on especially removing the IP barrier of genome editing tools, especially for global South, because there are we do not have, like from India and in this part of the world, the scientists do not have, most of the scientists do not have the freedom to operate, to use genome editing tools that are existing for commercial development. Yes, they can use it for research and development, like academic research and development, but for doing a commercial product development using this genome editing tool, they do not have freedom to operate. They have to buy license from the patent holder. So this POC1 has potential to at least minimize those dependencies. So let me introduce you to the genome editing fast. If you see, this is a crop genome book. Okay, so imagine you have a crop, and that is nothing but the genome has a 3 billion letter book, and you need to fix a single typo on page number 831. For example, it is shown here 831. So you need to fix a typo in this page, but this book is itself a very thick book. How would you do it? So with traditional, breeding technologies and traditional technologies that we have, it is very difficult to go to 831 page and fix that typo. But with the emergence of genome editing, we can do that kind of, specific and precise changes at a precise targeted position in the genome. For example, suppose the code says that I am functional. With genome editing, you could make it I am non-functional. So it means if you have a negative regulator gene, or if you have a gene that causes allergy, or if you have a gene that, that is a susceptibility gene that enhances disease occurrence, so you could just make them non-functional to get rid of this negative function or negative traits of that plant. So how it came to the biologist, because it came from a biological curiosity in bacteria while scientist was studying or curious to know more about bacteria, and then it actually gave rise to one of the most powerful biotechnology platforms of our time, and that's genome editing. So let me tell you how it came as a tool. If you see this, suppose this yellow box is a bacteria, you all know that bacteria is infected or they are attacked by bacteriophage or phage virus. So these viruses, normally what they do, they insert their genome inside bacterial cell. And some bacteria, they learn how to protect themselves from those virus infection. So what they do, those bacteria, they take a snippet of that genomic material or the DNA coming from the virus, and then they keep that snippet of DNA, viral DNA, as a memory in their specific genetic locus. That locus is called CRISPR locus. So, for example, here when a red virus attacks, so they keep the red DNA snippet here as a red box, okay? And this is called spacer. And this spacer is nothing but a memory of an old or past infection of a virus, okay? For example, if there is a violet or there is an orange or there is a green virus attacks, they also keep a small portion of DNA in that specific locus as a past infection memory. Clear? Now, when this memory locus is transcribed, they produces normally RNA, and these RNAs are called CRISPR RNA. And scientists have found that there are certain protein coding genes that are present just and that are sitting just beside the CRISPR locus, and those genes are called Cas genes, which is called CRISPR associated genes. So this one of the most well-known and well-studied gene is Cas9. So this Cas9 is nothing but a protein which has the ability to cut DNA. So, for example, this Cas9 inside a bacterial cell, they remain vigilant inside a bacterial cell, always like a police. Okay. So what I said until now, that CRISPR locus transcribe into RNA, which is called CRISPR RNA. And for each of the memory, for example, there is a red memory, there is a violet memory, there is orange memory, and there is a green memory. For each of the memory, there would be a separate CRISPR RNA. So there is one memory, second, third, fourth memory. It means this bacteria has been attacked by 4 different kind of viruses. Now, I also told you that there are certain genes that produces protein. Those protein, one of the protein is Cas9, and this Cas9 goes and bind with the memory RNA, and they form a complex. And this complex always active in for any incoming DNA coming from outside for any new viral infection. For example, they're like police. Okay, so this Cas9 forms separate complexes for separate memories, and there are 4 complexes. And whenever there is a new infection, for example, here the virus comes, a green virus, so this green virus injects its green DNA. So these complex, Cas9 CRISPR complex, they will find a homology with this green DNA. And once it finds a homology, it understands that this is a problem. Why this is a problem? Because it knows from its past infection. So this DNA has a homology. It means this DNA is coming from a virus, which is my enemy. And immediately the Cas9 gets a signal, and it will make a cut in this incoming DNA. And as a result, the incoming viral DNA would not be able to replicate. And replication does not happen, and then bacterial infection also does not happen. And as a result, this bacteria is saved from this viral infection. So this is how the CRISPR immunity actually works. Now, what we learned from this natural immune system of bacteria? What we learned, that there is a small sequence, the memory sequence in that RNA, which is crucial to define which DNA to be cut. Clear? So that's the learning that we have learned from this CRISPR immunity. Now, what scientists have thought, what if we replace this 20 nucleotide with our nucleotide of interest, which will be having a match with our gene of interest, so that we can target this Cas9 to that gene and to make a double-strand break. Okay, so this 20 nucleotide natural spacer would be replaced with our own 20 nucleotide of interest that will have a match with our target gene. So how it actually works? For example, this is a customizable RNA that you have designed as per your sequence of interest, as per your gene sequence of interest. And this is the Cas9, which is the DNA cutting protein I told you. So suppose, for example, this is a postman. This postman has 2 scissors in its pocket. And what you give to a postman? You give an address to the postman to deliver your cargo, to deliver your letter or anything. Okay. So here I call it as a postman Cas9, and this address I call it as a genetic address. So if you give this genetic address to the postman takes the address and it goes to the genome. And when it will search where is this address located. When it finds a similar sequence, and then it will bind there, and it will tell the postman that yes, here you need to execute your job. Okay, so here is the address now found, and now you execute your job. So what is the job of this postman? This postman has a job to make double-strand break, because it has 2 scissors. 2 scissors means it has 2 nucleus domain. This protein has 2 nucleus domain. So it will make a double-strand break. And as a result, the target gene will be non-functional because of this double-strand break. Now you can ask, okay, it is understood that you can make a targeted double-strand break, but how the editing happens? So here is the answer. So once the double-strand break occurs in your target gene, then cell takes it very, very seriously, because any DNA damage is really very dangerous for the host organism. Now it will immediately try to repair this break. And during repair or resealing the 2 broken ends, it can inadvertently introduce some insertion or delete some original bases. So this is so it means While religating the broken end, it can insert or delete. So this is called random insertion deletion or random indel. And this random indels actually causes the disruption of the open reading frame sequence of the gene. Suppose a gene is 1 kb or 1000 base pair in length, another gene is also 1000 base pair in length. So if I ask you what is the difference between these two genes, then you would tell obviously their sequence is different. Okay, so one sequence actually makes a gene unique or they're functional because of their own sequence. Now, if there is a insertion and deletion and it causes the open reading frame lost or disrupted, then this gene will not be able to produce a protein that is supposed to produce in its original form. Okay, so that's why a gene can be easily knocked out by designing a simple small guide RNA and giving to the Cas9. Okay, now I think this background is beneficial for those who are new to this technology because there will be a lot in this online platform. But all the genome editing proteins that we use today in our crop improvement program, those are derived from natural microbes, and those are derived from natural microbial diversity and their protein diversity. For example, Cas9, Cas12b, and there are a lot of Cas9 orthologs. TNPB that we developed from our lab, those are all derived from bacteria and other microbes. But these proteins, they often show notable functional trade-offs because they are imported into a non-native environment. Suppose eukaryotic is not their environment, so they can show a lot of problematic functional trade-offs. And now, because AI or artificial intelligence is impacting every field of human life, and biology is not an exception. And biology has a huge application of AI. So AI-enabled design, can provide a powerful alternative to this naturally available proteins. So to bypass evolutionary constants, so there will be a lot of evolutionary constants. Suppose a prokaryotic protein is not well functional in an eukaryotic system. And also it can generate editors with optimal properties, for example, less off-target, high on-target efficiency, and many other things. Now, I think all of you know this, okay? A large language model actually revolutionized how we write, especially the writing, okay? Answering anything, for example, the GPT models that you use in your day-to-day, like OpenAI, Meta, Gemini, Cloud, and Grok, whatever. My question is, what if LLM or large language model is trained on biological diversity? If the large language model is trained on biological diversity, so then this large language model would be able to produce biological entity. For example, they can design biological biomolecules from that trained data set. So this idea came into existence when this paper came from a group from Ali Madani and his colleagues. That was published in 2025. So what they have done, they have first collected a huge genomic data, especially the metagenomes from the microbes, from the human microbiome. So 26.2 terabase genomic data they mined. And I detected CRISPR arrays and capsids from those genomic data. And then they found CRISPR-Cas operons, like 1.25 million operons was found from this terabase pair of genomic data, and they made a CRISPR-Cas atlas. And what is a focused CRISPR-Cas atlas? So CRISPR-Cas, those are single effector CRISPR-Cas. It means, there are a lot of CRISPR system, if you go and read, and if you Google it, there are a lot of different types of CRISPR systems. So, and there are different classes of CRISPR systems. And some classes are single effector, some classes are multiple effector. The single effector are usable for genome editing for very easily. So, for example, Cas9 is a single effector system. Cas12 is a single effector system. So now, after building this CRISPR-Cas atlas, then they have separately built a trained a model, like large language model with universal protein language model they have built, and that they have named as ProGen2. And this ProGen2 was trained with huge number of protein database available in the world. And with this ProGen2 now is fine-tuned by additional data set from this CRISPR-Cas atlas. Now this ProGen2 is fine-tuned, is more powerful, and they know the language of CRISPR-Cas system, which is a single effector system, especially useful for genome editing. After that, these like it's now it is like a GPT, okay? So it can generate its own because it is trained with protein language model, so it knows the language of protein and the evolutionary language. It can generate its own protein sequence, which would look like a CRISPR-Cas, okay, which would look like Cas. And in the first go, 4 million sequences were generated. And 4 million sequences is a huge sequence, and now there will be a lot of screening to screen out unnecessary sequences and choose the best sequences that have similar kind of structural features with the natural, effective, and known and well validated Cas systems. Like predicting 3D structures and many other, biophysical properties of those generated sequences by the language model. And the outcome is novel, diverse CRISPR-Cas proteins with potential genome editing activity, and then it comes OpenCRISPR1. So this OpenCRISPR1 was released to facilitate broad ethical use across research and commercial applications for genome editing. So, for example, if I compare OpenCRISPR1 with normal Streptococcus pyogenes Cas9, which is the most widely one and for which the Nobel Prize was given to Emmanuel Charpentier, Jennifer Doudna. So it is 403 amino acid distinct from the SP-Cas9. So it expands sequence space beyond natural diversity, but it has all the catalytic residues retained, and also it has the ability to bind the DNA RNA interface, because this protein Cas9 must bind to the guide RNA. It also must bind to the target DNA. So it has 3 properties. Okay, one is it needs to bind with the guide RNA. Second is it needs to bind with the target DNA. Third is it needs to make a cleavage in the target DNA. So cleavage is another property. So this 3 property is retained or preserved this function, and they are structurally also more or less similar with the existing SP-Cas9. But the difference is 403 amino acid. So there's a huge difference from any naturally available Cas. But it has improved specificity, so less off target in nature compared to the SP-Cas9. So this OpenCRISPR1 has high potential to overcome the IP barrier for Global South, because it is released and it is proposed to be used as an IP-free tool for genome editing for commercial development. And our lab is also interested in developing alternative to Cas9 and Cas12 and other IP-protected tools as we develop TNPB also. So what question we have asked that, will OpenCRISPR1 work in editing plant genomes? And how efficient they are compared to natural Cas9? And can OpenCRISPR1 be adopted for advanced editing platforms, for example, like base editing and prime editing? I will introduce you to base editing and prime editing a little later. So this OpenCRISPR1, and we named it Plant-OpenCRISPR1, POC1, engineered for efficient function in plant cells. So what we have done initially for a pilot experiment, we have designed 2 guide RNAs and gave to Cas9 and gave to OpenCRISPR1 that we developed in our lab. And then simply we have done in protoplast to see whether they are able to make a cut at both the guide RNA position in the same gene. And if both the cut happens, like double strand break here, double strand break here, sometimes the intervening region could be deleted, and that could be detected if you run them in a simple agarose gel electrophoresis. So if you see here, this run here with Cas9 with POC1 Plant-OpenCRISPR1, you can see this lower band visible. It means it is able to delete the intervening region exactly as expected. Okay, so this is a wild type band, and here along with wild type band in case of Cas9 and also OpenCRISPR1, you see the lower band. It means there are some population where the intervening region is deleted, and we have did the Sanger sequencing of this lower band, and you can see here there are around 500 base pair deletion in the target gene as expected. So that was the first result that gave us very to pursue aggressively this work, because the efficiency is almost equal like Cas9. So that was a very turning point to go into details of this study. So then we have designed, several other targets. For example, SUIT11, SUIT14, and Z3. There are 3 genes in rice, separate 3 genes. In all the cases, we compared with Cas9. PRGB32 is a well-known vector that has the natural Cas9, and this is our vector which has the OpenCRISPR1. And you can see the OpenCRISPR1 and the SPCas9 have comparable efficiency, and all of them have a non-significant, although non-significant differences from the naturally available Cas9. And their mutation profile, you can see here, dot means deletion, and these boxes are the replacement, and boxes are the insertions, and this bold letter is the substitution. So it means it mostly causes insertion, deletion, and very, very few substitution. Now then we question, is it also suitable to it is also suitable to act with another trans-CRRNA or the scaffold that are present in. So we recently developed from our lab a novel trans-CRRNA. So what is trans-CRRNA? If you see a Cas9 and guide RNA composition, this is a Cas9, and the guide RNA actually composed of these 2 RNA. One is CRISPR RNA and another is trans-CRRNA. Now this trans-CRRNA, this green color, so mostly the conventional, in all the cases, most of the laboratory use this conventional small trans-CRS, because if you see Streptococcus pyogenes gene, they produces 2 different type of trans-CRRNA. One is small from here, and another is this larger trans-CR from here. So large one, this one, and a small one, this one. Now, small trans-CRRNA need to be always combined with CRISPR RNA. So this is small trans-CRRNA combined with small CRISPR RNA. It makes a scaffold, and that is suitable for genome editing. Now, if you use this long trans-CR or long trans-CR RNA, which was not known whether it would be able to do genome editing or not. So we have done that and demonstrated that this long form of trans-CRRNA could also do genome editing, especially in plant cell from our laboratory. So we then asked that whether this long trans-CRRNA can also be compatible with the OpenCRISPR1 system that is developed through AI. So you can see here, this is normal Cas9, the lighter one, and the darker one is the OpenCRISPR1. And we compared with all 3 variants of trans-CRRNA. One is conventional, and another is a long one, another is a deleted long one. So delta trans-CRL. So this is an engineered trans-CRL. So all these 3 cases, it shows that POC1 is compatible with these 3 different versions of scaffolds, which is actually expanding the toolbox. Now, So from this point, what you understand that POC1 exhibited editing efficiency comparable to SPCAS9, which is with the natural one, across all tested loci in rice with no, in most of the cases, no statistically significant differences. Now we have asked ourselves whether it could be adopted for developing a precise genome editing tool, which is an advanced in nature. Now, those are Indian audience and those who follow Bollywood movie, I think you can imagine from which movie it was taken. It is from Pa. And from this movie, this is Oro. So Oro is a 12 years old in this movie, and he was suffering from progeria disease. Because of the disease, he looks so old, and his friend is so young. And this progeria affected child normally dies at the age of 12 years or 13 years or within 15 years. And you know why they die at the age of this early age? Because they have a wrong DNA letter. Instead of G, they have a in their genome at a specific position. And as a result, Oro suffered from progeria and died at the age of 13. Similarly, if you see a crop perspective, this crop is susceptible to a disease because of a C nucleotide, whether this is tolerant to the disease because of a replacement with a T. So there's a single nucleotide polymorphism present, and that's why one is susceptible, another is tolerant. Now, my question is there a way to rewrite genetic letters? Because here, the normal knockout tool where, you give a guide RNA and it goes and make a double strand break, and then random insertion deletion occurs, and the open editing frame is disrupted, and the gene is no longer functional. That system does not work here. What do you need to do? You need to exactly rewrite A to G here. You need to rewrite C to T here to make a disease-tolerant plant. So the question is there a way to rewrite genetic letters? The answer is yes. You can do this precise single letter change by using a technology called base editing technology, which is developed from David Liu's lab from MIT. And how it works in the genome. If you see this is your genomic code and here is a problem and you don't see a phenotype in your genome, now you have a base editing tool and you could correct that genetic code. You could replace the wrong letter with a right letter and you get your phenotype back. So this is how the base editing works in the genome. Now, what we have built now, we have built POC1 mediated adenine base editing first, which has the ability to make adenine to a guanine, so rewriting from adenine to guanine. And we compared with the best version of adenine base editor in the market. And we targeted, more than one gene. And you can see this A was rewritten with a G nucleotide here. Precisely, A has been modified to a G nucleotide with this POC1. And if you see the comparison in one cases, it is better than the conventional base editor. In another cases, it is a little lower. So you can say it is comparable. But remember, this ability to modify A to G is not an universal case. It cannot make any higher A to G. A to G can only be made in a specific activity window. What is activity window? Suppose if you are designing a 20 nucleotide guide RNA spacer sequence, within that 20, it's not any higher, you cannot make A to G. There is a specific window where a base editor shows its optimum activity window. For example, the traditional base editor normally works 4th nucleotide of the spacer to like 4 to 8, very high efficiency, and modern base editor sometimes shows extended window up to 12. Okay. So, but here you can see the POC1 makes very good editing at second position also. But the second position editing was very nil or negligible in case of the traditional base editor. but the POC1 base editor shows very good efficiency at the second A of the protosphere site. So if you count the 20 nucleotide 1, 2, 3, 4 to 20, and then NGGPAM. So out of this 20, this POC showed an editing window range from 2 to 12. 2 to 12. But in case of normal base editor, so it is 5 to 12. So it has a little extended editing window. Now we also made another type of base editor that has the ability to change another base, that is C to T. So it can make a C base pair to a T base pair. So that's called cytosine base editing. So, and then we compared with the conventional one. So for example, you can see here how all the C's, this C has been modified to a T nucleotide. This is a wild type sequence. Here this C has been modified to TT. In all the cases with a very high efficiency. So if you see here the comparison, the light color graph is actually the traditional cytosine base editor, and this dark color bar actually shows the efficiency of the POC1 mediated base editor. But in this case, the POC1 showed significantly higher efficiency than the traditional cytosine base editor. And this is a comparison, in respect to the respective position in the protospacer, like C4, C5, C6, C7, like that, and across the 3 genes that we targeted and demonstrated. Now, let me take you to another advanced technology, which is very interesting and most advanced technology that allow us to do a lot of precise editing. That is called prime editing. Now, for example, until now, I have showed you one is a tool very specific that you can make a targeted disruption of a gene by making a double-strand break. But what kind of edit will generate from the double-strand break, you do not know. That's random insertion or deletion. Now, second technology I have introduced to is base editing. Now, base editing can do base substitution, one base to another base. For example, adenine to guanine, cytosine to thymine, okay? So single base substitution. But for example, imagine this is a situation, Tay-Sachs disease, where the affected child died at the age of 5. It's an inherited disease. And this disease happened because of a specific insertion, TATC in hexagene gene in human. So TATC is additional insertion, and that's why this child is suffering from this disease. Now, if I ask you, what is the genetic hypothesis you would make to cure this child? Now you would say, okay, this additional 4 nucleotides that are inserted, if it is to be deleted very, very precisely, and the original sequence could be restored, and this child could be cured. So that's an hypothesis. But with the technology that I have showed you, the knockout and the base editing, it is not possible to specifically, precisely delete exactly the 4 nucleotide. Because in this case, you cannot afford to delete 5 nucleotide. You cannot afford to delete 3 nucleotide. You have to delete 4 exact same TATC nucleotide deletion from the same position. Now, for this kind of editing in the genome, this technology is useful. Prime editing is a revolutionary technology also developed from David Liu's lab, which allow you to do this kind of all changes in the genome. And here, a little changes in the guide RNA sequence is needed. For base editing and knockout, you do not need to change the guide RNA. It's the same guide RNA. But here, this is a normal guide RNA or genetic address. But for prime editing, you need to use a little extended guide RNA, which is called prime editing guide RNA, where you have 2 definite sequence, which is very important. One is a search sequence, another is a replace sequence. Now, search is a sequence that will allow you to decide where to edit. And this replace sequence would allow you to decide what to edit. Where to edit and what to edit, both are coded in prime editing guide RNA. Okay, clear? So it gives you so much preciseness. But okay, this is the chromosome, this is the arm, this is the locus, and this is the base number. So this is higher, okay? Now, these 4 base need to be deleted, this 4 base need to be replaced, this 4 base need to be inserted. All this would be guided by this sequence, okay? So now let me give you a simple example. Suppose you have this problem. Now you have a prime editing. So what you need to do? You need to delete 3 and you need to insert 1. Okay, you could do with this prime editing. And you can get your sequence corrected and your original correct sequence restored, and you get your phenotype back. This is how it works. Now we build prime editing system with this Plant-OpenCRISPR background, and we targeted 3 different changes. One is C2G, another is C2A, another is G2C. So there are 3 different genes and 3 important single nucleotide polymorphism which are known for governing important trait. For example, ALS, ACC are important for herbicide tolerance, and this PEP-C is also very important for C3 and C4 carbon metabolism. And for example, for PEP-C, editing efficiency was insignificant, but in case of ALS and ACC, it showed good efficiency, obviously, but it was significantly lower from the original Cas9 based editing system, prime editing system. But interestingly, in one of the cases, in most of the cases, if you see the preciseness of the editing, prime editing is a tool, as I said, is very precise, but it could also sometime, like out of 100, it could also sometimes generate some undesired edit. And if you make a ratio of desired versus undesired edit, so if you compare that ratio in the 2 cases, PEP-C and OLS, non-significant differences between the ratio of desired versus undesired. But in 1 cases, ACC, the ratio is very high. It means it is highly preciseness. It showed high precise editing in case of ACC. But this is a smaller targets, only 3 genes, so there need to be a lot of genes to be validated to get a concrete idea how it is working. And we also generated edited lines by using this tool. And this is a generated plants, the knockout plants compared with this is a traditional Cas9 based vector targeting the gene CYP75B4, and this is a Plant-OpenCRISPR based vector targeting the same gene. We generated 25 plants. We only 25 actually screened and sequenced for data, and there is also a non-significant difference, although there is a little lower efficiency in POC1, but the difference was non-significant. So this work was published recently in New Phytologist, and this work is actually led aggressively by Priya and Romeo, 2 of my PhD student, about to complete very soon. They will complete their PhD. And if you need to design a lot of experiment for genome editing, and you need to design guide RNA, you need to design peg RNA, you need to design guide RNA for base editing. So many things are very complex, especially peg RNA designing is very complex. Now, after experimentation, you got the data, and this data analysis also another hurdle. So you need to have an assistance from some web tools or softwares and resources, computational resources to help you in those experiments. So, and there are a lot of those tools in markets and in literature, and then people are confused what to use, especially those are new in this field. So what we build a umbrella repository tool called CRISPR-Gate, where you have different modality of editing, activation, gene activation, gene disruption, base editing, prime editing, and those modality based tools and their ranking system, and we tell researcher how to use them and which one is being used by others very frequently, so people get an easy access to those tools through this repository. And this was done in collaboration with the ICAR-CRRI group led by Dr. Asif's group, and um, so this and this, if you suppose, for example, if you want to use POC1 for your experiment, for example, if you are in India, you can write to us, and you can ship this plasmid to you for academic use by simply signing an MTA. But if you want to use from other countries, so we have already deposited to AdGene, as we have also deposited our TNPP vector in AdGene, and there are a lot of people from different countries using our TNPP vector. Also, we have submitted these 4 main vectors that are very, very crucial, built with this open CRISPR system that are available from AdGene for ordering for academic purpose. And crop genome editing is making rapid progress worldwide. As you see, this is a world map recently made, and those green colors, including the Europes, also became green. They already approved genome edited crop that do not have any foreign gene inserted in the final product. They will be treated like conventional crop, and they will not be facing the stringent GMO regulation. So it's a rapid progress worldwide, and there are a lot of already commercialized genome edited organism across globe. For example, USA, Japan, and there are a huge number of crops. In the pipeline, they will soon be commercialized, and there are 2 from India. Also, there are 2 very encouraging results that 2 genome-edited rice varieties have been announced by government and identified for variety release. But there are some IP issues because they have used Cas9 and Cas12A. And hence, POC1 has the potential to further democratize this technology, especially in global south, and that is, we believe strongly. And I must acknowledge the very effective group and highly hardworking group. I think there are few of this, group members are still in the lab working, and these are the funding agencies that actually support my work in CRRI. And thank you all for this invitation. I think I'm on time. So back to you, Anil, and Sherjin, moderator. And I'd love to take any question.
Yes, sir. Thank you so much, sir, for the wonderful presentation. It was really captivating. So I request
Being taught, and I know you are a good teacher. Very nicely you communicate the things to students. Yeah, in this group there are many students and people from academia and some researchers from the industry as well. So now we are opening for the question and answer session. Any questions? I don't see any questions in chat. Yeah, one raised hand. David.
Yeah, you can unmute yourself, David, and you can ask the question.
Okay. I have a few doubts. Sir, you mentioned that base editing POC1 that greater beyond expanded to 4 to 2 base pairs. If it is a bigger window, there is a possibility of bystander effects. So how we can overcome the bystander effect?
Yes, you're right. So it is a bigger window then, but this window was calculated considering all the genes that we targeted. But if you see for one gene, so obviously when editing window is bigger, then the efficiency of bystander editing also increases, and that is a problem for base editing, obviously, that I understand fully. But those, for example, in many cases you would not have that much of, suppose for example you are using adenine base editor, so your target A is present at like 6th position or 7th position, and probably it is likely that you do not have also A in the 2nd position, A in the 12th position. So obviously, so that is a problem. Yes, if it is extended window, it has more chances, or it extends your applicability, at 12 position editing, at 2 position editing, but also it enhances the bystander editing efficiency also. Yes.
Hello. Yes. Can
Yeah, go ahead, please.
thank you, sir, for your informative session. Can So I want to know that I have two questions. I want to know that first is regarding can we use CRISPR-Cas technology for overexpression of certain genes?
Oh, overexpression. Yes, you can do. For example, there are different approaches. So let me tell you direct approaches, and then I will tell you indirect approaches. So direct approach is you can use, this is called CRISPR activator is a technology where the Cas is fused with a gene activator. Gene activator means it's a transcription activator. Okay, so if you fuse your Cas with a transcription activator and you design guide RNA for a promoter of your gene target, okay, so this activator protein will go to the promoter sequence of your gene of interest, and because Cas9 is going to your promoter and the fused transcription activator is also going to the promoter. So that's why there would be available transcription activator at the promoter region of your gene of interest. And it already has been proved in multiple studies that this strategy works in enhancing gene expression multifold. For example, in our TNPB paper also that I have not shared here, we have used TNPB to make a gene activator, and that enhances gene expression like tenfold over the base level of the expression of that gene. But one problem of this system is your gene activator construct should always be present in your cell. So it will be a transgenic approach. It would not be a non-transgenic. You cannot get rid of the plasmid or the construct that you're using. So the first generation you are getting the activation, and if you remove the transgene, then your activation will also go. So if you want to see a continuous activation of gene, you have to keep that construct in your cell, so it will be a transgenic one. So that's one strategy. Another strategy is you can do promoter editing. For example, there are examples of cis element editing. You could remove some inhibitory cis element, or if there is some repressor cis elements, you could remove them and you can enhance the gene expression. But that's a trial and error. You do not know if those editing would really enhance the gene expression or not. But there are at least 10 to 12 papers that showed effective cis element editing to enhance expression and fine-tune gene expression without changing the coding sequence. But you have to change the promoter sequence, cis elements.
Okay, thank you, sir. So one more question can I ask? I read about the CRISPR-Cas technology, like kit they have developed for diagnosing some disease in the land directly. So is it like how far it is successful?
For disease diagnosis,
Kit is developed.
Oh,
CRISPR-Cas.
yes. It is highly successful because there are a lot of, even during Corona, so there is two very good system developed in the US. One is based on Cas13 to detect RNA viruses, and on Cas12a to detect DNA, like those organisms that has DNA as their genetic material, those pathogen could be detected with Cas12a. So one is called Sherlock, and another is called detector system, and there are a lot of this CRISPR diagnostic is in place. A lot of tool already commercialized in the West, and many Indian companies are also working on CRISPR diagnostics.
Okay, thank you so much, sir. Thank you.
Yes, sir. We have few more questions in chat box. This one is from Souvik Ghosh from IIAB. Can base editors only do transitions, or it can do transversions as well?
Oh, yes. So base editor was known to do transition. First base editor came as C2T, like CBE, and then A2G, adenine base editor. That two are the mostly highly efficient base editor, and those are the pioneer work from David Liu's lab. But later, now base editor can do all kind of changes. there is an expanded base editing toolbox that's called GBE and then CGBE. So you can read, I have an article in Trends in Genetics, like base editor platform extends to base editor landscape extends to perform, transversion. And so now all type of base substitution are possible with base editor, but all the efficiencies, all type of base substitutions efficiency are not equally good, but you have to choose different platform for different substitution. It is not that you are using the same base editor for different substitution. You cannot. Adenine base editor for adenine to guanine. Cytosine base editor for cytosine to thymine. There are thymine base editor, TBE, for thymine to adenine, like this. Okay. There is CGBE, cytosine to guanine base editor. But the efficiency and activity window varies. So their application also gets limited because of their low efficiency on very narrow, limited editing window also.
Okay, sir.
So yeah, you could read some of the article that we wrote recently and you would get those information.
Okay. Thank you, sir. Another question from Sinchana Kashyap. Hello, sir. It was a nice presentation. I have a doubt regarding any off-target effects by the POC1 and any usage of POC1 for organelle genome editing.
Yes, so you see POC1 is a new thing. Okay, so we are published first for plant purpose, and we develop like prime editing, base editing, and knockout system. So yeah, it has not been used till now. For example, organelle genome editing, normally CRISPR-Cas9 system is very difficult to use for organelle genome editing. Normally, organelle genome editing is done with talent system. All the organelle genome editor, mostly like more than 95% of organelle genome editor are based on talent system. It's not based on CRISPR-Cas9 system. Why? Because in order to in order to edit organelle genome, you need to transport two things. One is your protein, that is Cas. Another is the RNA. Cas protein can be transported inside the organelle very easily by fusing a transit peptide or transit signal, okay? Transport signal, peptide. But RNA cannot be transported. People do not know how to transport effectively and guide RNA inside the organelle. So that's why CRISPR-Cas9 is not suitable for organelle editing. Yeah, you could do some bialistic gene transformation method to directly, insert RNA and protein inside the chloroplast, but that are by chance things, but those are not established yet. So that's my answer. Yes.
Yes, sir. There are 3 questions from David Edison. How is POC1 is comparable with the TNPB efficiency? Because size is similar. 4000 A or 400 base pair or 1200 base pair.
Yeah, I got it.
Yes, sir. What is your way for easy characterization of all construct, either transient assay or all are stable transformation?
Yes. Thank you, David. I think that's a very good question. So first question, to answer your first question, is that we have not compared the POC1 with TNPB1 because POC1's effective control would be a Cas9, like real control, real positive control on the system, because its size is same with the Cas9, and the OpenCRISPR1 was developed with the language model built with the Cas9 system. Clear? So it is more or less like the natural Cas9, not like the TNPB. So the TNPB is not a good control to compare with. So that's why we have not directly compared yet with the TNPB editing efficiency. We have only data of comparable efficiency with the natural Cas9. So your first question answer is done. Now, second question is, what is the strategy to quickly validate a lot of constructs? Yes. For example, for this kind of work, you need to validate a lot of constructs which is working, which is not working. So we do a transient assay, for example, protoplast. So protoplast is a transient assay, which gives you also to validate your construct in native genomic context, because protoplast is a live cell. It's not an outside in tube, it's not an in vitro assay, so it's an in vivo assay, but you can get the data very quickly, like within 10 days, you can finish the experiment and the sequencing time that you require. So we do protoplast assay. We have a very well-established protoplast system in our lab. And that is also published. If you want to follow, that is published in, you can see my publication in A Biotech Journal. So we have published the protocol also. So protoplast is our answer.
So which cloning strategy is very good in CRISPR working lab? Either restriction digestion based or gateway based or golden gate based?
No, actually, for this kind of work, this perspective, actually, from in my lab, especially, there is no specific cloning method preferred over others. It's a need based. Okay, for one cloning experiment, you need Golden Gate. For another, you need Gibson. For another, you need just simple restriction ligation. Okay, so it depends on the feasibility. Which one is the easier one? We follow for that cases. For example, in one case, I would use this Golden Gate. Another case, I would use Gibson. Another case, I would use simple restriction digestion and ligation. So it's not a simple, straightforward, black and white answer that I prefer this, but I do not prefer this. Okay.
Yes. So two more last questions. Jimmy John is asking that any cases of reversal of CRISPR edited genes yet similar to classical gene editing?
No, I could not get this point clearly, actually.
Yeah.
So reversal of gene
Any
editing.
cases of reversal of CRISPR edited genes yet similar to classical gene editing?
Classical gene editing. Okay, so probably you're saying classical genetics, probably. So, for example, if we do targeted editing, and for our case, we have not seen any reversal of edited sequences. But suppose if it is not evolutionary fit, for example, you have created a sequence, novel allele, but that allele is not preferred by plant and it is deleterious for plant. So plant to try to get rid of that allele, okay? But normal, the genome editing are done by scientists. Those are all beneficial for plant. For example, disease resistance, for any other purposes. And we select them, very precisely select them to keep them or keep them inherited. Okay, so if you follow those allele inheritance by marker assisted selection, you would ultimately fix them in the population. But obviously in future, suppose we do not have that data like 15 years 20 years of an edited lines yet to say that nature can reverse the editing that has been made by CRISPR-Cas9. So we do not have those kind of data, even in other literature from other groups also. Yeah.
Okay, sir. Thank you. This is last. We'll take last question from Dr. Balaji from Rasi Seeds. What about the editing of multiple targets using POC1? Is constitutive expression of Cas9 is sufficient for targeting multiple targets at the same time?
Oh, yes. For example, in our case, the plan that we generated with POC1, that was not a big multiplex, but we have done with 2 guides simultaneously, and it worked very well. Single guide means it's a single, okay, single editing. But when you are using more than one guide, that's all multiplex. So we used at the most 2 right now. So we can talk about 2 only, but it should be similar. With like 4, 5 guides, 6 guides, because Cas9 have been already presented and demonstrated very effective in editing even 8 to 10 guides together, like a multiplex editing in different targets, like more than 8 and 10. In different system, you have to use PTG system like polycystonic tRNA, gRNA system, and there are a lot of multiplexing system you have to use, and POC1 is compatible with the system. So those are absolutely okay. But we have not done yet more than 2. Only 2 we have done. Yeah.
Okay, sir. Thank you. I would like to ask 2 questions, sir. So we know conventionally use different proteins for editing Cas9 for a range of things. But how actually AI actually
Yeah.
can guide us in constructing different these proteins, nucleases, in a quite elaborative way. Another thing is that, so when we compare this POC1 with OC1, with the original one, so how does actually the POC1 is efficient in reducing the off targets? Thank you, sir.
Yeah, you see, OC1 was shown to have significantly reduced off-target compared to naturally available Cas9, SP-Cas9. Okay, so it is also expected that POC1 would also show less off-target editing efficiency. But remember, in case of plant, off-target is not a great problem, like the animal system. Why off-target is not a great problem? Because if you see compared to other genetic manipulation techniques, like for example simple crossbreeding, so it is more way precise. That's one thing. Another thing is that if you do a simple back crossing, suppose if you do a simple back crossing with your parent, this is your edited line. If you do a simple back crossing, then your background is purified by a single back cross. Okay, so the off target is not a great problem if you see on target editing happened with high efficiency and there is no phenotypic anomaly. And there is a simple check. Okay, so you can just predict the off target sites computationally and amplify them by simple PCR and do Sanger sequencing, and you can see whether your edited line has any off target editing or not predicted. Okay, but you can also you have option to do a whole genome sequencing to see how much percentage, if there is any off target editing. But what I see in case of plant, it is considered off target is not a great issue compared to plant. But it is likely that it will have also, but we have not analyzed the off target, and compared with the Cas9, any off target efficiency differences. That's your second question answer. But can you repeat your first question a little quickly so I remind myself?
This POC is entirely based on AI, which is helping us to design construct, right, sir? So
Yes,
how can actually AI play into role in designing these things? So that was my question.
Oh, yeah. What I said, because in my first few slides, I have showed that how it actually worked. So there is a language model that you have built. You have to build on the protein language, like a huge database of protein from the natural diversity of that protein, a specific protein. And if you could build a model, language model specific to that protein, and then that model can generate a new sequence. And that's a AI generated sequence. And now this sequence, suppose this model can generate like thousands of sequences. Now those thousands of sequences may not be active in cells. They may be simple sequence like and a simple generated sequence, and they may not make any sense, or they may not have the functional properties. So you have to screen out, like you have to screen those generated sequences by building their structure, like Bozuzar bioinformatics in silico tool, and you have to validate whether they are folding rightly, their biophysical properties. There are a lot of biophysical properties, their PLDTT score, and their all the alpha helix, beta pleated sheet, all are okay. Their distances are same, okay, and their solubility matrix is okay. Like all the biophysical parameters, those pipelines should it cross, and now you are satisfied. Now you are satisfied out of suppose 10,000 generated sequence, and you are satisfied with 1,000 sequence. Now in that 1,000 sequence, you cannot check 1,000 sequence in your cell, so you have to make 1,000 construct. Okay, that's a very costly affair. Now you can also rank them by different criteria. Okay, this is the rank, and you can rank like top 100 or top 50, and then you can check them in animal or plant cell line whether they're active or not, and then you can just compare their editing efficiency. And if you see there are 2 or 3 candidates that show really very good efficiency, and even show better efficiency than any naturally available variant, then you're good to go with that AI generated sequence to build a new protein. Okay. So that's the pipeline that I showed. Okay.
Yeah. Okay. Thank you, sir. I was unable to follow it earlier stage, so I could not get it. So now I am very thankful for you clearing the doubt. May I request Dr. Anil sir to inform a vote of thanks?
Yeah. So thank you so much for this very wonderful lecture. And this is mostly constituted with most of students. Yeah, this kind of very detailed, basic kind of thing was very much required for this group. And there were some people from the industry. They were also benefited from your lecture. We wholeheartedly thank you, sir. Thank you for accepting this invitation and giving this wonderful lecture in this evening, late evening. Thank you so much, sir. By this, we are
And thank you
closing this session.
Yeah.
Yes,
Thank
sir.
you very much for this invitation and best wishes to share genes. May you grow higher, bigger, and more stronger. Okay.
Yes, sir. Thank you, sir. Thank you so much.
Thanks.
So, in the end, I would like to thank Anil Kumar, sir, as well for making this happen. Thank you, sir. And we thank all participants, and we'll wind up it now. Thank you all.
Thank you.